Hi everybody, welcome back to Chem 131. Sorry for the delay, I had to eat. We're gonna get started here with proteins. Proteins are an important part of your diet. As you've probably noticed by now, since this is the last of the major biochemical compounds that we're going to be talking about, we've mostly been talking about the stuff that the body gets energy from. So we started with carbohydrates, then we moved on to lipids, and now we're at proteins. So all of these molecules are important for cellular growth because we need these components to build the cells themselves and also to build things like enzymes or macromolecules that provide energy like ADP and ATP. So today we're going to talk about the protein part of this. So we're going to cover enzymes and stuff like that towards the end of the chapter. Oops, I got to get this on the screen here. And so the first thing we got to do, and we talked a little bit about this yesterday when we were discussing the casein lab, is what the heck is a protein? How does it relate to what we've talked about so far? Well, proteins are just amides, it turns out. Fancy amides. Now, when you take this... Do I have a picture of that? Yeah, there we go. When you take this shape, which is like the simplest carboxylic acid and amine that you can get, and you chain them together over and over and over and over, you get a protein. So let's go back a little bit and talk about what a protein is. So a protein is a repeating polymer of amino acids, and an amino acid is shown on the next slide, but I'm going to actually call up a sheet here that has all the different amino acids that are available. Now, I put polymer in quotes because a polymer typically is a repeating unit over and over and over, whereas in proteins there are 20 different amino acid residues, and they don't repeat in a specific pattern. They repeat essentially what would be to the untrained eye randomly, I would say. But each amino acid has a specific purpose and properties. I guess purpose is not really the right word, but properties for sure. And those properties are going to control various things, such as what pH is it stable at, what shapes does it contort itself into, and how it interacts with its neighbors. And you might think, well, if it's a polymer, what does it care about its neighbors? But in proteins, it's actually very important. So let me get that picture really quickly, which hopefully I saved it. I was looking at it the other day. Amino acids chart. I'm just Googling it, so. I'm a professional. I can speak confidently, and I know how to use Google. That's basically all you need, right? Here we go. I don't know why this window capture is... Oh, interesting. Okay, yeah, that's exactly what I wanted. Thanks, OBS. Let me just make that really tiny real quick. All right. So, here are the 20 amino acids. No, you do not need to memorize them. That would be silly. And you'll notice that at the top of the amino acids, I don't know if you can see my cursor here. There we go. At the top of the amino acids, all of them have the carboxylic acid part and then the amine part. And then coming off of the chain, the 2-carbon chain between the carboxylic acid and the amine, is what we're going to call an R group. And the R group is just whatever the thing is coming off of it, we're going to correspond that to one of these amino acids that exists. All the amino acids have a full common name. So, for example, this is arginine right here. A 3-letter code and then a one-letter code, because biologists got lazy and then they got lazier even still. I shouldn't say lazy, I should say efficient, but but some of the one-letter codes don't really make sense. For example, glutamine is Q. I wouldn't remember that. Most of them do, but like tryptophan is W. Like, okay. Now, almost all of these just have this R chain. There's one specific one, proline here, that has a weird loop. I'm not going to make you memorize these, obviously. You can always look them up on a chart. And then furthermore, on an exam or quiz, anytime I refer to a specific amino acid, I paste the structure in there. But I do rotate it around sometimes, because what I do is I type it in ChemDraw. Because I don't want to draw these over and over and over. I just tell it, okay, give me alanine. And it does that. And then I paste that into my document, and then I upload that to Canvas for the quiz. So you might get a structure where these are just spun around in some random orientation. And so it's up to you to recognize what the R group is and where the amine end is and where the carboxylic acid end of the protein is. Now, on the previous slide that we had here, so let me get rid of that. I guess it would be the next slide here. Here's what our default amino acid template looks like. And so this R group right here, is going to be whichever group that we want. So this determines which amino acid. And I'm going to leave that picture just there in the background of, we'll just have it handy here, because we're going to refer to it later on. When we start talking about protein folding, looking at those R groups will be particularly interesting, because we can discuss like, oh, this R group lets us do that, or this R group lets us do this other thing. All right, so how do we chain a bunch of these things together? Oh, apparently there's my link for the amino acids. How the heck do I chain a bunch of these things together? I was hoping that it would show me a picture, but it didn't. So let me just get this out of the way here. Oh, Photoshop, come on. There we go. So I'm going to take... We're going to do that. And I'm going to take a second one of these. I'm just going to put a different R on it. What I'm going to do is I'm going to use the amine. I'm going to add it right here across this OH carbon bond. And so if you remember how we do amine synthesis, I'm going to call this one R1, just... We do it this way. So I'm going to go H, N, H. So there's my NH2. Here's the rest of my protein here. And we're going to swap. So I'm going to make water out of this, and then I'm going to make my amide bond like that. So this will just go away, because we don't really care about water. What we do care about is Maya being obnoxious. My goodness. I made it 10 minutes into class. I've already yelled at her today. And by yell at her, I mean talk to her like this. Just be like, oh, seriously, Maya. So now I've got this going on here. So I made this bond just now. And then I've got R2. which I guess actually would go like this. And then I've got C double bond O, and then this is another OH. So now I've connected two amino acids together. And I could connect another one on this end or this end. So I could just keep doing this. I can connect all the amino acids I want to by doing amide synthesis. Now, you might also recall that, wait a second, we never used amines and carboxylic acids because that made salts. How the heck does this work? Well, we have an enzyme to facilitate the reaction. Because you're totally right, it would make a salt, wouldn't it? I'll just pass over the proton. And in fact, we're going to talk about that in a second here. But because we have an enzyme, we can use amides directly to catalyze the reaction. We don't need an acid chloride here. We don't need an anhydride. We can just do this directly. That's not fair. The body is cheating. Well, thankfully the body's cheating. Otherwise, we would have acid chlorides in our stomach. That would be awful. All right. Now, as I chain more and more and more of these together, I didn't realize that the this just says down here below my head here, where you can't see. The amino acid proline is a bit different. It has a cyclic component to it. When we chain a bunch of these things together, I don't know why there's more stuff that's... I want to talk about chaining things together. Amino acids are chiral. Like, nearly all of them are L. You could write them as Fisher projections. I'm just not going to. So, in our amino acids, you're always going to see the H2N group on the left, because that's the predominant form that we just find in people and other living organisms. So that's cool and all. Okay, this is what I was expecting here. We're going to talk about chaining them together later in the chapter. I forgot we're going to talk about pH. Okay, so when I have something that has an amine and a carboxylic acid, I have an interesting situation. So this is obviously an acid because it says carboxylic acid. It's right there in its name. But what about this part? Well, amines are basic. And so what this means is that in a solution, this could pass its H plus over to H2O to make that. And then that could come around and give the H plus back to this. So I could pass my H plus ion to a water molecule, which will then carry it over to the other side of the molecule. Now, in reality, the odds of that happening are essentially zero. What will almost certainly happen is that my carboxylic acid will pass the H plus to water, and that water will just tootle off and bump into some other amino acid and protonate that. And my amino acid I started with will be protonated from some other acid. Because molecules move in straight lines, right? Until they hit something. The odds of them hitting something and coming straight back, very, very low. But suffice it to say that in an acid that would be saturated with amino acid, I'm going to start protonating water with the carboxylic acid side. And then the base side, the amine side, will start accepting protons as it sees them. And so I will end up with a molecule that has two charges on it. The left side, the basic side, has accepted the H+, and the right side has lost the H+ to gain a minus charge. Any molecule that has two charges present on it that are opposite of each other, such that the total charge cancels out, is called a Zwitterion. So a Zwitterion has charges present, but overall is electrically neutral. And proteins are the classic example of Zwitterions. Now, in the casein lab, that's important because we can use the Zwitterionic form to precipitate out proteins. We just have to find the pH at which the protein we're looking for is ionic. It has both charges. And at that pH, the solubility of the protein goes way, way, way down. And then we can just filter it out. Now, most other proteins will become very soluble in water when they are in the Zwitterionic form. It takes very specific shapes and orientations to crystallize out proteins. Usually with amino acids, this is really trivial. You could crystallize an amino acid pretty easily. But for larger scale proteins, it's a lot more difficult. And we're going to see why this is when we talk about structure. But Zwitterions help us explain, why is it soluble? Well, it's soluble because things that are charged interact with water much more easily than things that are nonpolar. So that helps us dissolve our amino acid or our protein into water. Now, obviously, the Zwitterion can only form at the very ends of the protein. So if I have a really, really, really long protein, depending on what the protein's side chains are, it may or may not be soluble in water. And again, more on that later when we talk about structure, because solubility is a very, very important feature of proteins. But insolubility is also just as important. My goodness. I'm just going to keep it on my desk here and reach for the squirt bottle every time I see her out of the corner of my eye. Alright, now, what happens when I have a solution that already is low in pH? There's already tons of H+ available without me having to move my H+ off of the carboxylic acid. Will interesting things happen at various pHs? And it turns out that they do. So if I go over here and I draw, we'll get rid of that. We'll draw a generic amino acid. And I'm going to draw this twice, so I gotta boop, copy, paste. There we go. All right, so what we're going to do now is we are going to go low pH and high pH. All right, at low pH, we would consider an environment to be acidic. And in something that is acidic, lots of H+ is present, right? And so what that means is that I basically have H+ floating around everywhere. Now, again, this will be carried by water. And so we have two questions that we want to address. First of all, will this happen? And second of all, will this happen? For the first case over here, we can say, well, certainly this is going to happen. Eventually, the base will find our H+ and accept it. That's what bases do, right? Excuse me. The second one is a little less certain, though. Let's talk about what happens when you deprotonate something. I'm just going to call this R. This is my generic carboxylic acid. You might recall from Unit 10 in Chem 121 that when acids dissociate, they protonate water, so we would get this. Well, let me just write it this way. So these are essentially the same thing, H plus and H3O plus. And then we get the carboxylate form. However, Le Chatelier's principle says, well, if I already have tons and tons and tons of this in the solution, I would push the equilibrium back in this direction, right? So the predominant form of the carboxylic acid would be this one. So if I lower the pH by adding, like, say, HCl to the solution, then I have so much H plus in there already that all of the H plus then repopulates my carboxylic acid, and I end up with the protonated form again. So the conclusion that we would draw for our amino acid up here is that if there's already tons of this stuff available, this doesn't happen. In fact, the reverse happens. It tries to protonate this so that the OH remains in place. If this H pops off, another one takes its place, because there's just so many. So the form that we see at low pH ends up being, let's just erase some of this extra stuff. So we keep the OH as is, and this H2N, wait, what? I was drawing on a different layer? Oh, because I pasted, look at that. This H2N becomes H3N with a plus, and this OH remains the same. All right, let's look at high pH. What happens at high pH? At high pH, you can think of this, well, first of all, it's basic. You can think of it as either having very, very little H plus or lots of hydroxide. Now, if there is very little H plus, then this reaction is just not going to happen, right? Because there's just no H plus around for it to occur. So the amine side of our amino acid is going to remain unprotonated. It'll just stay how it is. But this side over here, this H will pop off because if there's tons of OH minus here, these two will react to make water. So what that means is that this side becomes this. So at high pH, the carboxylate side is deprotonated and has a minus charge. So that's this right here. I'm on the wrong layer. There we go. At low pH, the amine is protonated. Now, somewhere in between here must be this Witter ion. So if low pH, I can't actually draw there because there's a window in my way. If low pH is some number and high pH is some number, there must be some intermediate pH where both ends have our charge and we get our Zwitter ion. And that zwitterion will pop up at a specific pH, and that pH is called the isoelectric point. So every amino acid has an isoelectric point. And the pH at which this occurs depends on the R group. Depending on what other features are present in the R group, that may facilitate the deprotonation of one end or the protonation of the other end, depending on what's there. For example, if the R group is also an acid, then the amine side is going to get protonated pretty quickly. And so the isoelectric point would be at a much higher pH than it would otherwise. Now, it's not worth memorizing isoelectric points because those are all tabulated, and you can look those up whenever you want to. So that is what I would recommend if you need to do that outside of this class. For our purposes, I'm going to tell you the isoelectric point in a problem if you need to know it. So I might say, like, okay, at pH 5, this amino acid is at its isoelectric point. What would it look at pH 8? Or what would it look like at pH 2? And then you'd have to pick from a series of pictures, or you'd have to describe it, or something like that. So that's where we're going with pH. So pH is very important for our protein amino acid residues. Because later on, what we're going to discover is that, well, if I want to destroy a protein and make it so it doesn't work anymore, I just change the pH a lot. Changing the pH will ruin the isoelectric point of all these things. And it will also screw up various forms of protein folding. So pH is something that is very, very sensitive. Proteins are very sensitive to it. So why the heck do we have all sorts of stomach acid in our stomach, which is the very first thing that proteins you consume come into contact with? Well, it's because we want to denature the proteins in that case. We want to destroy the proteins so that we can break them down into amino acids so you can eat them. Not eat them, you've already eaten them. So you digest them and you can incorporate them into your body. All right. So again, the isoelectric point is when the pH is just right, and that allows you to have your zwitterion. All right, so let's look at some examples here. Well, we already talked about this. So here's glycine. Glycine has an isoelectric point of pH 6.06. If I lower the pH to 4, what will it look like? Which side will change? So I'm going to label the sides here. I'm going to highlight this and highlight that. You can tell me which side changes. So this is side A, and this is side B. Which side is charged at pH 4, and which side is neutral at pH 4? So type in what you think the answer is in the chat, and we'll discuss. This is mostly so that I can get somebody to talk in the chat. Because it's so quiet and lonely. All right, we got side B. Side B will do what? Can you be more specific? If you're an 80s or 90s kid like I am, side B is the side where all the crappy songs got put, right? There's technology that will have a real hard time explaining to younger generations. Yeah, and you rewind the tape when it gets all screwed up with a pencil. You just stab it right through this hole, right? Cassette tapes are weird. So what does side B do? It will become... Side B will gain an H. Very good. So, because the pH is low, there is abundant H+. So this will just hop right on here. Side A will remain unchanged, and this is what we get. Excellent. All right, let's do another one. This is still glycine, but now we're going to put it in at pH 10. So now what happens? What will happen to which side? And this is still side A, and this is still side B. So what's... oh, excuse me. So what's different about this scenario compared to the last one? Instead of having H plus everywhere, now we have OH minus everywhere. And so there you go. Side A will lose one of these H pluses to make a water. We don't really care about the water, but it's neat to know that that happens. And when it does, the left side, the amine side, will be unprotonated. The right side will remain the same because there just isn't enough H plus in the solution to find our carboxylate again. All right, so down here, I have two different amino acids. So on the left, I have alanine, and on the right, I have phenylalanine. And this is really just to get you used to drawing structures. So what I want you to do is draw each of these ions as a zwitterion, assuming its like isoelectric point is about 7, and it is. It's like 6 point whatever. And then tell me what the amino acid looks like when it's at pH 2, and then tell me what the amino acid looks like at pH 12. So you're going to be drawing these on your paper. So obviously you can't just draw them in the chat, not with ASCII art or something like that. So when you're finished, let me know. And if you have any questions, also let me know too, of course. So we'll go over the answers in just a little bit. So the formula that determines pH is, and this is from Chem 121, but it's kind of a weird formula. So pH is minus log base 10 of how much H3O plus there is. And so this is in moles per liter. So a solution with pH, well, I should say, so like if I have a concentration of this where there is 1 times 10 to the minus 7 moles per liter, which is a super, super small amount, I could put this number into this equation and I'd get minus log that number, and I would get 7. Whereas pH 1 would be 0.1 moles per liter. So it's a logarithmic scale. Every time you multiply the amount of acid by 10, the pH goes down by 1. So whatever acidity pH 7 is, there's going to be some H plus in water. If I have 10 times as much acid as water, then it's pH 6. And if I have 10 times as much acid as that, then it becomes pH 5. And then so on and so forth. I'll let y'all keep working. I just that was a question I couldn't answer just by typing. Do you want to say hi? Say hi. Say hi. You bashful now? Say hi. There you go. That's the other cat. You guys haven't met Kresnik because he never comes out. I'm not going to scare him away because he's shy. Oh, but Maya now immediately comes in. Here, you can come in. It's okay. She's looking at me with like, I don't believe you. Here, come here. Come say hi. Come say hi. Say hi. You tell them how upset you are with me because I keep squirting you with water. Because you keep bugging me during my lecture. Alright, I'm going to pet the cat, and then for one more minute, I'll let you finish these problems, and then we'll go over the answers. All right, let's go over the answers here. So let me clear off. Oh, that's weird. So you can see everything I drew on the wrong layer here. So we'll select this layer and get rid of all that too. Delete layer. Deleted. All right, and I've already forgotten, oops, which amino acids. That's not what I wanted. That's also not what I wanted. That's what I wanted. So I want alanine. Let me just scroll this up here. And phenylalanine. So all right, so first let's do the zwitterion. So the zwitterion would be this goes away. And this becomes a positive. Wow, that really doesn't look like a minus. There we go. And then at low pH, the amine would be protonated, and so would the carboxylate. So if we go to pH 2, this end stays charged, and this end gains a proton. And then if we go to pH 12, both ends are essentially deprotonated. All right, so there's our answers for alanine. And then for phenylalanine, really the only difference that I need to make here is I just need to do this. And you'll notice that I put the double bonds in different spots. It doesn't matter at all. There you go. So there's our structures. And that's what our amino acids will look like at various pHs. Any questions, let me know. All right. Now, what do amino acids do? Well, amino acids link together to form amide linkages, as we have mentioned already. But all of their side chains, the R groups, can have various things on them, which can facilitate any reaction that we've talked about in this class so far. That's not what you want to hear during week 10, right? Or maybe it is, because that means you don't have to learn anything new. You just have to apply it, right? Now, specifically, we're only going to look at a few particular reactions. One very important one, and then a few other just like, incidental ones. So this is not as scary as it actually could be. So let's look at the common reactions that we're going to do. So the first one is cysteine. So you might remember that if I have a thiol like this right here, there's my thiol, and I oxidize it with another thiol, I do this sort of thing, right? And so what ends up happening is when I oxidize thiols, I create a disulfide link. And that's shown here. Now, cysteine is the amino acid that has this on it. But this conjunction here that we have, this little bridge, this is called cysteine. So cysteine bridges hold two amino acids together. Now, if you can imagine a situation where I have a bunch of amino acids chained together, and one of them happens to be cysteine, and then somewhere over here, another one happens to be cysteine, you could imagine a situation where I could connect these two together. For example, if my amino acid chain went like this, right? And so the purpose of these cysteine links is that they allow the protein to be bent into a particular shape. And again, shape is going to be very important later. Now, if I reduce my cysteine by adding hydrogen, for example, I can undo the disulfide linkage. Hydrogen, not super common in your body. And when I say that, I realized that, like, wait a second, that sounds preposterous. And then I started yawning. So, awkward pause there. When I say hydrogen, I mean hydrogen gas. You do not have H2 gas in your body in large concentrations. So this is not so H2 is not a particularly effective way of reducing disulfide bridges back into amino acids. Not something that you need to worry about happening in your body. Now, when I react my two amino acids together to form my amide linkage right here, I create a peptide. So a peptide is just the result of when we take two amino acids and hook them together. And you could do this in any orientation that you want to. So for example, I connected this right here so that I got a sequence that went R1 and then R2. But what if instead I wanted to do this? So I took this one and I wanted to put it over here. Would that be the same thing? I would get R2 and R1, but can't I just reverse them by flipping it over like a sheet of paper? Well, it turns out, remember, these are chiral. So if this is pointing out of the page, and then I were to try and flip the whole thing over, they would now be pointing into the page. So the order that you do these actually matters a lot. So the order matters. You can't have symmetry in proteins because the group the R is coming off of is chiral. So these are all chiral carbons right here. So chiral, 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 chiral. And the orientation matters, obviously, like we've said. Now, when we chain a bunch of these together, you get a protein. Now, because there are 20 different choices that you can make for every amino acid, to make a protein that is one amino acid long, you would have 20 different combinations. If there were 2 amino acids, you would have 20 squared combinations. And if you had 3, you'd have 20 cubed. Just in case you were wondering how many choices there are, for just 3 combinations, or a combination of 3 amino acids, there are 8,000 ways to do that. And if you get to proteins that are fairly long, we'll say, like insulin. Insulin is fairly long. It's 51 amino acids long. There are 10 to the 66 different ways you can do this, and the body makes the correct one every time. How the hell does everyone beat the odds and make insulin? correctly? And the answer is, well, it's coded into DNA. DNA is written in a sequence of chemical pairs, and that particular sequence says, oh, do this, pair these amino acids together every time. We're going to talk about that very briefly in chapter 19, what the molecules are for DNA. And now my cats are fighting. Stop fighting. Stop fighting, meow. They're bored. Now he's talking to me, so now it's Kresnik who's in the hallway. All right, so we'll talk about this when we get to base pairs. So we get to adenine, thymine, guanine, and cytosine. But our purpose in the proteins chapter is going to be looking at the result of putting all of these proteins together. So now that we know the basics about proteins, when we come back from the break, because I don't know what my cat is screaming about, we're going to talk about how we fold proteins and how we can explore their properties and then get them to places where they need to be. So I'm going to put, not that, that's not what I want to put up there. Well, let's put this up on the screen. We're going to take our 10-minute break, and I'm going to go deal with the cat. And we'll put more Persona 4 on here. All right, almost end of the quarter, so let's find something that reflects that.
They fight all the place in it. Counted by the times that just how the life goes by. I know it's not your going to say, but there ain't no other way. So step on up to the place. Need to face what can spread on into the light of us. Step on up to the place. 'Cause this ain't no game. It's time to make history, yeah. It's a war out there every day. You can't hide from me. You gotta pray for the rules, pretty cool, calling out in the face of the savage scream. When your friends and boss look alike on the face of it, they miss the history, but they fight all the place in it. Yeah, they fight all the place in it. Counted by the times that just how the life goes by. I know it's not your fault you say, but there ain't no other way. So step on up to the place. Need to face what can spread on into the light of us. Step on up to the place. 'Cause this ain't no game. It's time to make history, yeah.
Alright, we're back. You know, there's only one more lecture after today. Weird to think about that. We are almost done. It sure still feels like there's a long way to go, which is funny because one of the songs that I played was literally called Long Way. But we're really right at the end of this, so we're going to do protein folding today. And then Maya's going to complain some more. No, she knows better now. I looked at her sternly and she walked away. So we're going to do protein folding today. I'm going to leave enzymes and enzyme activity for tomorrow because that plus the I keep calling them amino acids. They're base pairs. The DNA synthesis stuff is like 2 minutes. I'm going to show you the base pairs, show you the connection, show you the block diagram, and then you're done. So this will be the last section we cover today. We'll do enzymes tomorrow. So now protein folding is actually pretty complicated because proteins themselves just have so many different possibilities that they can enact to create new and interesting shapes. So let's start at the basics. We'll start with the sequence. We're not going to draw pictures of proteins anymore because that's just going to get super tedious. So this is what we're going to do instead. And even then, I'm not going to do this very often. So if I link 3 amino acids into a chain like this, my polypeptide of 3 could be represented like this, gly-al-val. Now, I could also use the 3-letter codes from, I got to get the amino acids chart again, which timed out on me. There we go. So I could get this, and we could give these a 1-letter code. So glycine would be G, I bet. I can't even find it. It should be in the... There it is. It's right there. Yeah. You can't see my cursor. So here's glycine. So I could write GAV for that particular sequence if I wanted to. And when biologists are writing textbooks and want to just type in like a sentence for common pairs of proteins that just repeat over and over, they just type that. So, all right. Now, when I have chains that include cysteine, remember that cysteine can make a disulfide linkage. I can make a new particular shape, the hairpin that I talked about before. So whenever you see cysteine in a chain, that's going to be one of the interesting things that we're going to be looking for. Other ones we'll look at on a case-by-case basis, but cysteine is one of the particular important ones. All right. Each protein that we form requires a certain number of amino acids. And that's going to depend on what it is and how complex the material is that we are creating. So insulin only has 51 amino acid residues. Hemoglobin is much, much larger. It's 574. And it also has parts of the parts of the structure aren't even the protein. So the protein is only part of hemoglobin. There's also the porphyrin ring and then the iron ion that is used to carry oxygen. Now, the point here is that you saw the rough size of a molecule when I put 3 of these together. Imagine putting 500 of them together. It doesn't matter how you twist or contort this, you're going to get something that's quite large. Because proteins are really big, we don't really have the ability to move them through cell membranes very easily. So if you remember the pictures of the cell membranes from the lipid section, there is always like the protein transport duct that was shown in them, and so you could slip some proteins through there, but not giant proteins like hemoglobin, for example. That's very difficult. Instead, what we can do is we can pass through individual amino acid residues pretty easily. We could get those to where they need to go. So cell walls and cell membranes and things like that restrict where proteins can be once they're assembled. And so proteins tend to remain within cells to do whatever it is that they need to do. Now, what does a protein specifically do for you? You have lots of proteins both in your body, coming out of your body, and those are probably the two main things, right? Inside and outside of your body. So inside, what I mean would be things like muscles. So let me just put all of these things on the screen here. So your muscles are structural support for your bones. They help provide mobility. When you move any part of your body, like when you stand or when you wave your arms around like one of those inflatable car salesman things with the fan at the bottom and they go woo. If you're doing that right now, you know exactly what I mean. Those require muscles for you to move your body, and proteins are what make up muscles. You also have other structural features in your body, such as things like your fingernails. Your fingernails are made of keratin, and those are proteins. I think they're made of keratin. Is that right? I'm going to have to look that up. Your hair is a protein, believe it or not. Yeah, fingernails are made of keratin. Okay, I just want to make sure I didn't get that wrong. What else do you have for proteins? As far as like simpler proteins are concerned, well, I guess they're not simpler, but they are smaller. So like hair is just a really long polymer protein. Other things proteins do, so like hemoglobin is used for transporting around oxygen in your body. It also can remove carbon dioxide by the same mechanism. Other proteins in your body would be things like insulin, enzymes that catalyze various reactions. Yeah, things like that. So proteins are everywhere. And that's why it's very important that you have a source of protein in your diet. So you can get proteins from plants. You can get proteins from animal meat. But we, generally speaking, can't create our own proteins in our body. And the reason for that is because we don't digest nitrogen super easily. Proteins are our source of nitrogen because they are compounds that contain nitrogen. Carbohydrates don't have nitrogen, and most lipids also don't have nitrogen. The other source of nitrogen would just be the air around us, but we don't fix nitrogen in our stomach or our lungs. So we cannot breathe nitrogen and then turn that gaseous N2 into a protein. Our body just doesn't have the capacity to do it. All right. The order in which we put all of our proteins together will dictate the shape that the protein will eventually take. And the shape of the protein will be crucial when we talk about enzymes tomorrow. Now, the other feature about shape is that shape dictates surface area of a molecule. And the surface area is what things around the molecule interact with. For example, water. If I drop the protein into water, sploosh, it's now in the water. If the protein has a very high surface area, and along that surface area are places for water to interact, it's more likely to be soluble. Whereas if the surface area is very low, and there are fewer places for water to interact with, then it will be insoluble. Depending on your properties, you may or may not want your proteins to be soluble. For instance, your muscles, probably good that they're insoluble in water. If you put a steak into water, it doesn't immediately dissolve. That would be terrible, right? Like, not only for like your steak, because like you'd be eating mush, but also if your muscles dissolved every time they're exposed to water, you wouldn't have muscles at all. You would just be goop, right? You'd be like the slime creature from that ancient movie in the 40s or whatever that my parents watched growing up. The Thing or something. So structure is really important. So those particular proteins are insoluble in water. On the other hand, hemoglobin is transported through your bloodstream. We don't want the hemoglobin to fall apart, obviously, because then we couldn't transport oxygen. But if it was completely insoluble in water, we couldn't carry oxygen through the bloodstream to places it needed to go. So there needs to be some degree of solubility there. And the shape and the groups that are attached to the outside will help dictate that. So let's start at the beginning. We're going to look at just a particular chain of amino acids and then zoom out from there. We're going to discuss 4 different structures, and these are classifications, not like 4 different things. So the first one we'll talk about, which is primary, is the most zoomed in, and then we're going to zoom out to secondary, and then we're going to zoom out to tertiary, and then finally we're going to zoom out to quaternary. So that's what I want you to think of this in terms of structure. So the primary structure of a protein is just whatever the order is for the proteins that I've chained together. So for example, this sequence that I wrote on here, glycine, alanine, valine, and leucine, has some particular function and some particular shape based on what those R groups are. And those R groups are, do I still have this page? Yeah, here we go. Again, there's those R groups. So glycine doesn't really have an R group, it's just a hydrogen. Alanine has this methyl group. Valine has an isopropyl group. And then what would leucine be? Oh, here's leucine. Okay. So these are not particularly interesting functional groups because they're all alkanes, right? But alkanes still can do dispersion, so that might be interesting. And we'll talk about that when we get to tertiary structure. So primary structure is just the order in which the molecule, or which the proteins, the amino acids are sequenced. When you put enough of those together, you can then use various properties of the amino acids, not the R groups, but just the amino acids themselves, to get things to stick together. And the shapes that we observe are called secondary structures. There are going to be 2 shapes that we're going to want you to recognize. And then the third one we'll just attribute to secondary structure, which is not really a shape, it's just random. So now, what holds secondary structures together? Strictly speaking, we are looking for hydrogen bonds, but the hydrogen bonds are not from R groups. These hydrogen bonds would come from the amine, the NH, and then the C double bond O, that O. We're going to point the NH towards the double bonded O on a different residue. And that will make a hydrogen bonding interaction. If we get enough of those together in close proximity, we can hold stuff together pretty tightly. Now, unfortunately, this is kind of the best picture I could find. But as you can see here, let me draw over this with my pen. So here's my nitrogen. Here's the hydrogen connected to it. Here's C double bond O. And I can point that hydrogen in such an orientation that I can make these links between different parts of the protein. I'm just trying to find them so I can circle them. There we go. And what ends up happening is if I do this in a particular way, I get this sort of spiral. And so this spiral is called an alpha helix. And what an alpha helix is... Now I have to spray bottle the other cat. I don't know what's going on today. Probably the construction. They're renovating next door and they're probably distressed. So here I'm spraying with water. Where was I? Alpha helices. So when you swirl the swirl the amino acid chain around like this, you provide opportunities for the proteins to line up over other proteins. And when they do so, they can form these hydrogen bonding interactions, and this holds them into this little coil. Now, in this circumstance, there is one principal chain that is coiling around itself. Now, keep in mind that this is a little bit different than the double helix structure of DNA, which has 2 strands. This only has 1 strand. So this strand is attaching to itself to hold itself together. Now, in the second instance, if I draw my protein sort of like the snaking pattern, and I have parts of the protein going past each other, what that can look like is this. So here is my... I have the wrong pen selected here. So here's my nitrogen, here's my hydrogen, and then my C double bond oxygen. And I can make hydrogen bonding interactions with the residues on the other side of the protein chain after it is lapped around. And so this looks kind of like a sheet that I have folded on top of itself over and over and over. And so we call it that. This is called the beta pleated sheet. Why is it a pleated sheet? I don't know. Pleated sheets are kind of obnoxious to work with. But that's just what they call it. And so naturally, when I wrote this slide, I called them ribbons. But really, if you think of it like a long contiguous sheet that you're just kind of letting it fold back and forth on top of itself. I'm moving my head to make the motion, but you can't see my head. My head going back and forth as if that's me putting a sheet and just going back and forth to fold it or something. But these types of interactions are actually very common because the identity of the amino acid is more or less irrelevant. As long as the R groups don't get in the way such that they push these protein chains too far apart, you can form these hydrogen bonding interactions with any proteins or any amino acids that you want, except for proline. Proline is the weird one. Remember that proline here, shown on the very far right, the shape is a little bit different when we've made the amide linkage, and so that H there won't be present, and so you can't hydrogen bond with proline. Proline would be a hydrogen bond acceptor, but it would not be a hydrogen bond donor. So if you have a bunch of prolines, that might mess up the shape. But if you put them, like, say, over here or something, or maybe over here, you know, where they can't get in the way. Sorry, proline. And then finally, the last secondary structure that we care about is just the random structure. It's not really a structure because it doesn't have hydrogen bonds necessarily involved, but maybe 2 parts of the amino acid could hydrogen bond for just a little bit, and you end up, instead of getting like a spiral, you just get something that's like that. But whether or not hydrogen bonding is involved from the amino acids themselves, any part of the structure that just sort of coils about randomly in space, we will call it a random structure. You might see sources call this a random coil or something like that. A random coil would just be, oh, it doesn't coil enough or efficiently to make it look like an alpha helix. All right, so those are the 3 secondary structures. So we've got alpha helix, we've got beta pleated sheet, and then we've got random. Now, how are you going to be able to identify these? Well, you're just going to have to check them off of a list for one. Or if I gave you a picture, for example, you might have to tell me where the proteins are. Now, what this picture is trying to communicate, a lot of proteins look like this, where instead of showing the molecule, they just show you this loop, kind of like a path that the chain of the amino acid follows. And you could see some of the other molecules over here. Other molecules that are important to the structure are usually shown, but for the remainder of them, so like what are the R groups that are coming off of here? They're just omitted. So whenever you see something that just looks like, so this right here would be a random structure. Over here, a random structure. This part right over here, that would be a random structure. Those are all random because they don't really seem to make any appreciable shape. But you can see here that when we make this really wide part of the ribbon, here's another one right here, and then here's another one right here. Those are all alpha helices. Now, the protein does not need to do that for very long for it to be categorized as a helix. Like 1 or 2 revolutions is good enough. And when you look at the chain, it will be very obvious that, oh, this thing is looping around itself to get into a specific shape. Then we have the beta-pleated sheets, and you can see that if you follow the arrows, because the arrows are shown which direction the protein chain goes. So for this orange path, it goes this way. Then it loops around and comes back and goes right parallel to it. And then earlier on in the sequence, something is parallel to that too. And so our hydrogen bonding interactions, let me get a different color for this that would stand out more, could be like right here. So here between the proteins, and then between these two, they'd be like this. And then in the background, you could see this light blue and this light green one. They could have hydrogen bonding interactions right here. So that's what these proteins look like when they are folded. And when I say protein folding, that's what I mean. The protein would be just like this long chain of amino acids just all strung together. But when I allow it to loop around randomly, eventually the interactions that are strongest will form. And it turns out that for that particular chain, this is the most stable result. This is the result that forms over time. Now, what is this? I don't actually know what this is. This is just a picture that I found of a protein that was small enough that I could identify the fragments of it. But you're going to see these fragments in bigger proteins. I have a hemoglobin coming up when we talk about quaternary structure, and you're going to see all these again. Now, when I take all of my structures and I put them all together, the result that I get, so this whole thing is the tertiary structure. Now, we do not count the secondary structures as being responsible for the tertiary structure, primarily because you could have a straight line chain and get an alpha helix, like it would just kind of twist upon itself, and then you could loop it back on itself and get a beta-pleated sheet. But why do I get this particular orientation? So why does this protein go all the way around and then loop around like this and then come over here and then loop back around to do that? What makes it do this turn or this particular turn? And the features that make it have that structure are the tertiary structure. This turn right here is probably because I have a cysteine disulfide linkage. Or maybe I have molecules that are interacting through R groups right here. And that causes them to attract enough that I can then bend this into the correct orientation that I need to. So there are going to be a bunch of tertiary structures, and those tertiary structures occur between the R groups. So this is where the R groups come in. And so these shapes arise because the R groups provide additional connections. Now, only in the in the case of cysteine, are they actual bonded connections. The rest are interactions. But we're going to explore a bunch of different ones. So the first one is the disulfide bridge, which you've already seen. So the disulfide bridge is the tertiary structure. Here's our disulfide bridge. If I do this, can you still see the sulfur? Not really. There we go. So having that disulfide linkage forces my protein to go and make a sort of U-turn. And if my protein were to try and like pull apart, it can't because these disulfide linkages are holding it in place. Now, these will complement hydrogen bonds. So this would be a secondary structure right here, because this is a hydrogen bonding, not between R groups. But these right here would be a tertiary structure. So disulfide bridges are probably the most common tertiary structure that we're going to refer to. They're the most obvious one. The next ones are not as obvious because they involve specific R groups to be present. So, for example, if I have an R group that has a charge on it, and I allow that to get next to another R group that has a different charge on it, I can create an ionic interaction, and this is called a salt bridge. And I can do this between any two functional groups that have ions on them. So let's go back over here, and we'll point out which ones those are. So anything up here in category A, anything that has a positive charge can interact with anything that has a negative charge. Now, on histidine, you might be thinking, well, where is the positive charge? You can put another H on, I want to say it's this one right here, because this is a secondary amine. So you could put an H there and make that positively charged. Arginine and lysine just generally have a charge based on pH and things like that. Over here, these are both carboxylate groups. Because they are carboxylic acids, they will deprotonate at specific pH. And then we'll have a negative charge. So I could pair either one of these with either of these three to make a salt bridge. So if I have an arginine and then a glutamic acid somewhere later, I could curl the protein around and get those two R groups to point at each other. I can make a link there, and that link will hold the protein in that shape because that attraction is strong enough to do so. So salt bridges are a tertiary structure that is possible depending on what R groups are present. I think the next one's either hydrogen bonding or hydrophobic. Yeah, hydrogen bonding. Okay. Do I have a picture of that too? So I don't like this picture because they're showing hydrogen bonding, but this is also ion, kind of a salt bridge. So first I'm going to just highlight hydrogen bonding occurring between R groups because that's what we want to know. But then I'm going to go back to here, and we're going to find the groups that can hydrogen bond with each other. And so those are anything that has OH or NH on them. So anything in category B. So this has an OH on it. This has an OH on it. If I got those two across from each other, I could hydrogen bond from one of these to one of these. There is an OH group in category D, tyrosine over here, and so you could make a hydrogen bond to that too. But primarily, we're going to be looking at stuff. Oops! Oh goodness, that's apparently the next picture that was in this slide. What else is in here? I mean, this just makes me think of like avocados because I see guacamole, but we'll go back to this. I just found this website somewhere, so I accidentally clicked on it. But these OH groups, not this NH2 up at the top here, because that's the amine part of the amino acid. That's going to be bonded to the next amino acid when we make our peptide. But these specifically, these groups down at the bottom here, this NH2 and this NH2 could form hydrogen bonding interactions. And then finally, we have hydrophobic interactions, which is basically, oh, I have a bunch of chains that have nothing polar on them at all. And this is a really shitty picture, unfortunately. But in this structure, I have nothing here that is polar at all. It's all nonpolar. And so what ends up happening is I have dispersion forces that hold those parts together. So it's more or less the same mechanism by which lipid tails hold each other together. So if I have a bunch of saturated fatty acids, their tails are all linear and would hold together. Now, if we go back here again, there's a whole category down here, category D, of hydrophobic side chains. And so all of these things down here are essentially nonpolar, or they are at least large enough that they are mostly nonpolar. So you can imagine if I had a bunch of leucines and phenylalanines all next to each other, the only thing they have is dispersion, but they are relatively flat molecules, so I could just sort of stack them together and allow them to interact. So those are the 4 types of tertiary structure. So let's just put all the summary stuff in here. So primary structure is the order. The secondary structure is from hydrogen bonding on the backbone of the amino acid. So that's either alpha helices or beta pleated sheets, or we can write random. And then finally, the tertiary structure comes up from interactions between the R groups. And those are either going to be from cysteine, so disulfide bridge, from anything that has a charge on it to make a salt bridge, anything that has OH or NH2 on it, or anything that has just nonpolar groups. So those are our 3 categories of structure. So far, we have quaternary structure to discuss, which is just take your entire protein and then allow other proteins to stick to it. So, and that's just, let me show you a picture of that here. So, this is hemoglobin. And I have colored the hemoglobin in, I haven't colored it, somebody else has. I've done 2 red areas and 2 blue areas, and they're kitty corner from each other, or cattywampus, or whatever you want to call it. But this chain and this chain are not connected. They are independent, and one terminates on both ends. So like this residue right here ends here and also ends here. It does not start as a connection to the blue thing. The blue thing is independent. However, there are interactions that hold these 2 things together. And those interactions, which are all the same interactions we just talked about, so they could be disulfide bridges, they could be hydrophobic interactions, they could be hydrogen bonding, they could be ion bridges, whatever. Those make up the giant macrostructure of multiple proteins that we see. And that is the quaternary structure. So the way that we're going to define quaternary structure is that when you have multiple polypeptides, and those polypeptides are held together in the same ways that we just talked about, so all the tertiary structures, we call that a quaternary structure. So tertiary structures hold, or tertiary interactions, I guess we'll say, hold together a single peptide. Quaternary structures are when multiple peptides are held together, as you can see here. And so you can see from this, we have gotten to the whole zoomed out picture by starting with just the sequence of amino acids as they were written in, then seeing how the chain itself could interact with itself, and then seeing how the R groups could contribute to making unique shapes, and then seeing how the entire thing is held together by those same interactions to make a giant macrostructure. So that's how proteins fold. So you're going to be asked about the nature of these interactions. Like I might ask you to check all of the secondary structures that are shown in a picture. Or I might ask you, like, from this list, tell me da 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 da. Like which of these is which, or categorize them, or something like that. All right. Now that we've learned how to fold proteins, how do we unfold them? Because when we consume proteins, say for dinner, we don't want our proteins to remain folded. We want them to unfold and hydrolyze and break them down, right? Well, because all peptide bonds are amides, you can hydrolyze proteins to turn them back into amino acids and then use them for whatever purpose. Well, guess what your stomach has? It has the substance that you need to do that. So if you hydrolyze a protein in the presence of strong acid or strong base, we'll highlight that, you break down the protein into amino acids. Now, this requires a little bit of heat to do. In fact, if you want to do this to your meat to soften it up a little bit, this is why you would cook something like, say, in a slow cooker in the presence of an acid. So, for instance, if you are slow cooking pulled pork and you want it to fall off the bone, you don't cook it for 10 minutes. It won't hydrolyze enough, and the proteins will remain bonded together, and you'll get meat that's really, really tough to eat, to bite through, and it probably won't even be cooked all the way through, right? So, what you do when you slow cook is you cook for a very long time at low heat, and then you add something acidic. So, most people do vinegar, like apple cider vinegar and a bunch of spices will assist in breaking down the proteins to the point where the meat becomes soft and tender. Now, if you're eating meat, whether you've cooked it to tenderness or you've cooked it to be a steak or whatever, when you chew it, you are breaking down the proteins by force, but you're also providing a base. So in your saliva is slightly basic. That helps assist the breakdown of proteins while you're chewing. And then when you swallow it, your meat, that's vulgar. Redact that. When you swallow your chicken or your pork or whatever, God, that still sounds bad. When you digest, it goes to your stomach, and then the HCl takes over because there's lots of acid. Wow, this got really inappropriate really quickly. Your stomach has HCl in it and will continue the digestion process and hydrolyze the protein further, so that by the time that it gets to your intestinal tract, those amino acids can then be transported through your bloodstream or to wherever it needs to go. All right. So that's the most basic way of breaking down proteins, is by hydrolysis. But there are other ways to do this too. For instance, if you add extreme conditions into the mix, so heat the heck out of it, raise or lower the pH to a very high or low point, what ends up happening is you interfere with tertiary and quaternary structures. And so you end up getting a really interesting situation depending on the structure that you're looking at. So cysteine bridges, you have to reduce back down, so you need a reducing agent. So acid or base, not so great for that. But hydrogen bonding, if you protonate stuff and make it charged, you change the interaction. If you protonated, say, the carboxylate group, you would remove the charge on it, and that would ruin your salt bridge. So changing the pH or changing the nature of the solution that the protein is in causes its structure to change, and its folding will either become something different or it will become random. And this is called denaturing. When you denature a protein, the structure is randomized. And when the structure becomes randomized, the protein loses its function. Because the function of the protein is, it needs to be soluble at this temperature and this pH and whatever. And it needs to have this shape so that it can accommodate this size molecule and whatever. If you destroy that, then it's just a sequence of molecules stuck together. A good example of this is egg white. So if you heat eggs and you look at the egg white, it's not really white, right? It's clear. So this is called the albumin. The egg albumin is transparent, and it is a bunch of proteins that are soluble in that medium for a specific purpose. And that purpose is when the egg gets fertilized, the yolk provides so I don't know the exact biology that's going on here, but the yolk is where the chick develops, and then I believe that the egg white is just nutritional support and other stuff that provides energy for the development of the fetal chick. But that needs to be soluble so that it can pass through various membranes and be used when it needs to, and so on and so forth. So it's a protein that's in a very particular configuration. But when you heat it, you essentially randomize all of the connections in the albumin. And what ends up happening there is, if you have randomized connections, it becomes much, much less soluble. And so the end result is that you see it visibly. It becomes white. You get a solid that precipitates out. That solid is then edible and is also delicious. So eggs are yummy, and I eat them all the time. Now, you don't just have to do this with heat. You can do this with other things too. For example, some people like their eggs over easy. I like my eggs with 6 molar HCl. So let's show that. I don't eat your egg with 6 molar HCl. Like, don't be ridiculous. So this is 6 molar HCl being poured into a solution of egg white. So the egg white's already in there. We're going to dunk the HCl in there. Here we go. And what you can see when this happens is that it instantly cooks, quote unquote, the egg. And so that looks a lot like when you've just started cooking your egg, right? Like, it starts to congeal and sort of come together. HCl will do the same thing. What's happening when you do this is that you're denaturing the protein. Now, the other purpose that denaturing proteins serves, in addition to just hydrolyzing things, is that it destroys the proteins to the point where they can't do their original task. And this can be really important. For instance, if you have bacteria in your food, and everything has bacteria, right? Bacteria everywhere. You have bacteria in your body. All of your food has bacteria on it. Why do we cook it? Well, one of the reasons we cook your food, or our food, I should say, is to denature that bacteria. When the bacteria's proteins get denatured, they're still there, but they're harmless. So cooking your food to a specific temperature will ensure that the bacteria that are present are denatured. And so even though there's things like salmonella in chicken, salmonella can't hurt you if you've denatured all of it. The protein is still there, but because its function has been randomized, it will no longer have the particular function of causes you to get food poisoning or get extremely sick. Now, you can do this with other things too. So, for example, this slide in particular would be if you were going to an in-person lab, we would be making you wear goggles. But at the end of the day, you put your goggles in this cabinet, and then when I leave the laboratory, the last thing I do is I close the goggle cabinet and I turn on a UV sterilizer. What UV light does is it's very high in energy, and it causes specific bonds to be denatured and ruptured, particularly double bonds. So if you do this with bacteria on goggles, obviously I'm not going and cleaning every single pair of goggles. So all the bacteria from the previous person are still on your goggles, but they're dead and they're harmless, so we just can ignore them safely and we can reuse the goggles. So this is an effective way of sterilizing stuff. You may have seen UV or even radiation treatment for food in various contexts. So, for example, at the grocery store, some specific veggies may have been irradiated or UV irradiated to prevent the growth of like bacteria or fungi or things like that, so that they just last longer. I grew up on a farm when I was a kid, so this is why I talk about food and stuff like that all the time, because that was what we did, was we produced food. Stuff that you pick does not last very long once it's off the vine. Only a few days at most, and so you need to preserve it in some way. Typically, the way to preserve it is you put it at very low temperature. But another way that you can preserve it is you can just sterilize it with some sort of UV or something like that. And then the bacteria on it are killed, and it takes much, much, much longer for the food to decay. In that period, we can then transport it to the store, whereby you then purchase it and then eat it. So denaturing proteins is just as important for us as a civilization as understanding how they fold and function. Here's something that's really relevant. Dun da da. Who's waiting for the vaccine? It's me. I'm waiting for the vaccine. How do vaccines work? Well, bacteria and viruses are just a bunch of proteins. If you denature them, they lose their original function. But your body isn't that picky. It is that picky in the sense that if the protein is in a certain orientation, then it will do harm. For example, COVID-19 is a viral protein that has a particular shape, and when it interacts with the human body, the shape of that virus is how it interacts with cells and other things. Now, your immune system can intercept the COVID-19 virus by looking at the particular shapes of its surface features. So, for example, you've probably seen the blobby picture. That's a really bad blobby picture. So everybody's seen the COVID-19 like cartoon picture, which is, it looks like this, and then it has these like spikes coming out the sides, right? These spikes are proteins. And so what we can do is we can create a denatured spike protein. And even though it has the same primary structure as the viral protein, it doesn't have the same tertiary structure because the protein's been denatured. Then what we can do is we can administer it to a person, and the body goes, hey, I know what that sequence of proteins is. That's bad. And the immune system will then attack that sequence of proteins, and it will remember, okay, if I see this specific order of proteins, I want this gone. And so then your body knows, okay, I can deal with this specific order. Then when it sees it over here, it's like, oh, I know what to do. Boop. And you're rendered immune. Or at least resistant. So this is the premise by how vaccines work. Vaccines are really effective because they are denatured viruses or bacteria. And so it's a template that your body can work with. I'm so jealous, everybody who gets to get there first does the vaccine. I'm like at the end of the queue because college instruction apparently is not considered essential, despite the fact that they want me to go back to work in the fall. And I'm like, get me a vaccine or I won't do it. Move me up the queue or I won't do it. Like, not okay. I'm not going to be in a room with 48 people who may or may not have gotten the vaccine. Like, don't be absurd. In a classroom where there's no ventilation and no windows, like, no. I have choice language for those people. But if it isn't clear, I would strongly recommend that everybody get the vaccine because it's safe, it's effective. We have data to show that it's effective. And I'm teaching you right here how it works. It's a denatured protein. And so if your body is exposed to a denatured protein, it sees the sequence, it sees the primary structure, and can recognize that and terminate it. But when it sees the actual protein, even if it's in a tertiary structure, it will just look for the primary structure and remove it. And then you're immune. All right. So there are all sorts of different reasons that we would want to denature proteins, and we use them in medicine. We use them in culinary stuff. I didn't know where to take the word culinary. Culinary deeds, food. We use them in food. All right. Now, we talked about heavy metals a long time ago when we talked about sulfur. What do heavy metals do? Well, if we have these heavy metals and they find salt bridges or cysteines, they tend to get in the middle of those. So, for example, mercury would hop right up next to the carboxylate group and be like, hey, buddy, how you doing? And then that salt bridge would break down. Or it would hop between two sulfurs and change the structure. It would make it longer, and that would cause the protein's shape to change. And the result is it denatures. This is obviously really bad. When you ingest compounds that contain these heavy metals, they tend to interfere with proteins very, very quickly, and that's bad. Many of those proteins are essential to body processes because they'll be like enzymes and stuff. So other stuff that we're going to look later on that does this is stuff like arsenic, or arsenate, basically. The problems that these run into is that they fit into enzyme space, and then the body's like, oh, I have this thing. I can't get rid of it. Now I can't use this to do whatever I'm supposed to do. And that enzyme is no longer effective. And then you don't get the material that you need for wherever it needs to be. That being like specific, like you don't get succinate or malate or whatever in the right spot of your body. And that would obviously be very bad. So heavy metals, don't eat them. Don't eat mercury, don't eat lead, don't eat silver, don't eat cadmium. Like, just avoid. Specific heavy metals are important for your daily diet. For example, you do need iron. And the reason you need iron is because that is specifically one of the metals that helps with oxygen transport. There are other reasons for other metals too, but I just don't know them off the top of my head. I can tell you what they do outside of the body, but I don't know what they would do inside the body. So, like, I take a multivitamin every day just because, like, why not? It's like $20 will buy me 6,000 of them, so I'll just take a multivitamin. Why not? What's the worst that could happen other than me peeing it out? I don't care. But inside it, it's like, oh, you're getting your daily dose of vanadium. Like hell, I know what vanadium does for the body, but all right. And that's all we're gonna do today. I think that's all the time we have. Let's see. Oh, look at that! Right on the dot, 2 p.m. So we're gonna save enzymes for tomorrow. Enzymes and then DNA. That's really all there is left to this class. There is almost done. So I want to thank everybody for coming to the lecture. Tomorrow we will finish up the last of the material. I will then unlock the last quiz. And then we'll talk about exams. Because exam number 2 is going to be everything since exam number 1. And right now my gut is that I am going to have exam number 2 on Monday, just like I did for the last one. And then the final exam will be Wednesday or Thursday, whichever one works for you. Because I realize it's finals week. And that other people have other finals that they may be taking. So we may have to schedule stuff. So we'll talk about that more tomorrow once we're done, because we'll have time to do that. But until then, enjoy your evening. And I will see you tomorrow for the last lecture. Take care, and I'll see you then.